a quick run through again so that we're on the recording. We start with our mouth to our esophagus, through our stomach, to our small intestine. At our small intestine, we have a couple of accessory organs that enter in with the liver, the gallbladder, and the pancreas. And then we go to the large intestine. So we get our food, and then it goes through our GI system, and it comes out as stool. All right, so this is us eating our food. It travels through our mouth, through the pharynx, esophagus, stomach, small intestine, and comes out your anus as stool. All right, first organ up, area up is the oral cavity, so your mouth. Here we have breakdown of the food mechanically and chemically. So mechanically we have mastication. Mastication is you physically chewing the food with your teeth. And then chemical breakdown is with salivary amylase, which is an enzyme produced by your salivary glands. Here are the three salivary glands that we have. You have your parotid gland, your submandibular, and your sublingual. They produce liquid fluid that helps lubricate your oral cavity and produces enzymes to start breaking down the food that you're eating. So you could say that digestion begins in your oral cavity. Okay, so today we're actually not going to be looking at the salivary glands or the oral cavity of the pig, so we just wanted to show you a quick picture. They sit relatively in the same spot as they do on a human, so your parotid gland is anterior to the ear, and then your submandibular gland is below your jaw. Okay, after your mouth, we have the pharynx. The pharynx is a point of connection between your nasal cavity and your oral cavity, so it's very important in breathing. It's also important in swallowing, and it enters into the esophagus, which we will show a video of swallowing your food.
Swallowing, or deglutition, is the process by which food passes from the mouth through the pharynx and into the esophagus. As simple as it might seem to healthy people, swallowing is actually a very complex action that requires an extremely precise coordination with breathing, since both of these processes share the same entrance, the pharynx.
Okay. So right where it's encircled is your pharynx, and then we will continue on to, click off it. We'll continue on to the esophagus. So the esophagus is the long tube that transports food from your oral cavity down to your stomach. It has an upper esophageal sphincter and a lower esophageal sphincter. Sphincter, you can consider them as circular muscles that tighten in order to close the tube. So the upper esophageal sphincter is closed when you're trying to breathe. It opens when you're swallowing. And then your lower esophageal sphincter opens when the food wants to pass into the stomach, and then closes to prevent food from exiting the stomach and entering back into the esophagus. Okay, next up, the food will enter into your stomach, where your stomach will secrete acid and enzymes to continue to digest the food, to continue to chemically digest the food, and also it'll churn food to enhance digestion. So this image, the stomach just looks like a hollow sac-like organ, but in actuality, in the muscle, in the walls of the stomach, there are muscle layers that contract to do the churning of the food. And then on the inside wall of the stomach, you have rugae, which are bumps and little hill-like structures that just increase surface area. And within the rugae, well, actually, the rugae means folds. Within each rugae, there are little openings that secrete the enzymes. So the food travels from the stomach into the small intestine. So the small intestine receives the chyme. Chyme is digested food, or starting to be digested food. In the small intestine, the major action will be absorbing nutrients and vitamins from these chyme. Small intestine is made up of the duodenum, the jejunum, and the ileum, which we will go through each right now. So the duodenum is the first portion of the small intestine. So it's the first time that the chyme enters into the small intestine. It receives enzymatic secretions and bile from 3 accessory organs. Your liver up here, this bright red, this dark red organ, your gallbladder, which is the green one, and then your pancreas, which is the orange organ right there. All of these 3 organs secrete their bile and their enzymes through ducts, which are tubes, and they enter into the duodenum through the ampulla of Vater. So I've said bile a couple times, and I'm sure you've heard it before, but we figured we would tell you exactly what bile is. It is an alkaline fluid that emulsifies lipids, which means it physically breaks apart fats. It's made in the liver and stored in the gallbladder, and then after you eat a really fatty meal, the gallbladder contracts and it is secreted into the duodenum through the ampulla of Vater to digest the fatty foods. So here is where your liver, this is where your liver anatomically sits. So it sits on the right side, upper right side of your abdominal cavity. And it is in charge of filtering the blood from the digestive tract, and it detoxifies all the chemicals that we ingest, and then also secretes bile. And you can see all the tubes where the bile comes from the liver. It could go into the gallbladder, and then from the gallbladder and the liver, it can go down through the common bile duct. So here is your gallbladder, which we have seen before. It is where bile is stored and concentrated. And like we said before, it contracts to release the bile into the duodenum. We have a case study. We have a couple of these throughout the presentation. So gallstones are a concentration of digestive fluid and bile that hardens within the gallbladder. And the symptoms that you get when you have gallstones is really, really intense pain in your right upper area, in your right upper quadrant of your abdomen. It can also create nausea and vomiting. You can get diagnosed with an ultrasound. So this is an image of an ultrasound that was taken of someone with gallstones. So this dark oval shape is the gallbladder, and then these 4 small circles in the gallbladder are your gallstones. And there's numerous ways you can treat your gallstones, but if you have gallstones that are filling your entire gallbladder, like this image, you could do a cholecystectomy, which is a removal of your gallbladder. Okay, next up we have our pancreas. This is the 3rd and final accessory organ that works to secrete enzymes into the duodenum. So it will again use the ducts to secrete the enzymes into the ampulla of Vater, into the duodenum. That's part of the GI system. It also produces glucagon and insulin to regulate your blood glucose levels. Glucose is another way of saying sugar. So we have a second case study, and this is pancreatitis. Itis just means inflammation, so this is inflammation of your pancreas, and it is when the pancreatic enzymes start to digest the pancreatic tissues. This usually happens because there's some type of occlusion, so a blockage in the ducts of the pancreas or into the duct that enters into the duodenum, and that way the digestive enzymes can't be secreted into the duodenum. So instead of going into the small intestine, they stay in the pancreatic tissue and they start digesting the pancreatic tissue, which is not good. Common causes are alcoholism, gallstones, smoking, and a fun trivia fact, scorpion sting. The symptoms would be abdominal pain so severe that you feel it all the way radiating into your back, nausea and vomiting, and weight loss. We diagnose pancreatitis using blood tests. So we're looking for those enzymes that were supposed to be secreted into the duodenum. They are now going to be elevated, and so we'll see that on blood work that we take of someone that has pancreatitis. So to treat pancreatitis, we would treat the underlying cause. So if we had gallstones and it was severe enough, we would do a cholecystectomy. Smoking, we would try to get the patient to stop smoking. And a scorpion sting, we would do the antidote for that scorpion sting. So just a quick review, because I know it just was a lot of information. After your stomach, you have your duodenum, which is the first portion of your small intestine. There are three accessory organs that secrete bile and pancreatic enzymes into the duodenum: your liver, your gallbladder, and your pancreas. They all enter through the ampulla of Ureter. Okay, so after your duodenum is the second portion of your small intestine is your jejunum, and the third portion is your ileum. These two portions of the small intestine are responsible for absorption of your nutrients and vitamins. Your jejunum is the main absorption site. So it's going to absorb the majority of your fats, carbohydrates, proteins, and water to get good nutrition and vitamins. And down here at the bottom of the slide, we're going to talk about calories per gram for 1 gram of fat, carbohydrates, and protein. So for fats have the most calories, and carbohydrates and protein are the same with 4. And what we're trying to get at here, and hopefully Kel can help me out if I misspeak, but your body needs so much energy to perform daily activities, and we get our energy from our calories. So we have 9 calories per 1 gram of fat, 4 calories for proteins and carbohydrates, and however, we only need so much energy. So the carbohydrates and proteins are usually a substantial amount of energy to perform our daily activities. We can use fat, but fat tends to have more calories, and if we don't use all the fat, all the calories from the fat, it becomes stored, and that's when we don't use the fat, and it just becomes stored throughout our organs and on our body.
Fat is the most effective way to store excess energy in your body.
So within our small intestine, within the lumen of our small intestine, on the inner wall of the lumen, are plicae circularis. Today I'm not entirely sure if you're going to get a good image of them on the small intestine of the pigs, but these are mucosal folds that contain villi, and villi's purpose is to absorb anything and everything that passes by it. So we have plicae to increase the surface area so that we can get more villi in our small intestine for maximum absorption. Next up, we have our large intestine. So I'm just going to show you right here, if you can see the cursor on the screen, you see the end of your ileum. So like we said, the small intestine is a giant tube that sits in the middle of your abdominal cavity, and the end of that tube goes into your large intestine. So that area right there is called your ileocecal opening, and there's a valve in there called your ileocecal valve. So our large intestine is consistent of our cecum and our appendix at the beginning, and then it travels up the right side of our abdominal cavity as the ascending colon. It cuts across as the transverse, and then right behind and to the left of the stomach, it turns down to be the descending colon, then the sigmoid, finally into the rectum, and then where the stool comes out, exits our body, the anus. So we're going to walk through each portion of the large intestine. So first up, we have our cecum. Our cecum is the beginning portion of our large intestine. It's a blind pouch. So there's only one way that your chyme can go from your small intestine into your colon. It has to go up through your ascending colon, where it starts becoming stool. So we will now show you that there is an opening to the appendix. So your appendix is lymph tissue, so it helps with your immune system. There is an opening which leads to our next case study, appendicitis. So the fecal matter that travels through your colon can sometimes get into that opening of your appendix, and that's how you get appendicitis. Okay. So the main responsibilities of the colon, which is the ascending, transverse, and descending portions of our large intestine, is to remove salts, water, and nutrients to form stool. Now, just keep in mind that even though it is still absorbing water out of the fecal matter, the jejunum is still the main site of absorption. So majority of your water is still going to be absorbed from your jejunum. And then your rectum, the last part of your large intestine, is going to hold the stool coming from the colon before it is released out of the anus. Okay, so now we're going to dissect.
All right, time to get to the exciting stuff.
Oh, not this. Okay, can we all hear us? Hope so, because Kale's going to show you how to dissect now.
Okay, hopefully everyone in the labs can get some assistance from those already there. Do you guys have your pigs in place? I'm guessing not. So the first thing you need to do is put your dry sheet down underneath the tray. Make sure you don't spill anything. You'll have your metal tray that you should be able to sit your pig inside of. So before we actually start cutting, we're going to want to stabilize our pig in the tray. So hopefully you've got some string or twine of some sort that you can use on your site. And so you will tie one side of the string, so you can see here. So tie one of the legs on one side, then wrap the string around and underneath the tray, and bring it around to the other side to tie the legs and keep them spread open. You want to do that to both the front and hind limbs to keep them in place. So this will probably take a few minutes. If you guys have any questions,
type them in the chat.
Yeah.
And then we'll be able to hopefully answer you. We'll give you a couple minutes to be able to tie your pigs down. I just wanted to gauge to see how everyone's doing with the tying of the pigs.
Everyone got them wrangled down yet?
That's gonna make a really corny joke. But bet you didn't think you were coming to a rodeo today. Okay, so micro lab is ready. Some of them are like,
So we're still going.
they're ready. They seem ready.
We're still live.
They seem like they're about to be ready. I think we did fetal pig. I did frog in grade school. My frog was pregnant. Have you ever seen a pregnant frog? I'm gonna Google it for you because like their eggs are like just out in their abdominal cavity. It's this, so that's an absurd. Okay. Alright, so let's get started. I think we get started. Everyone say they're ready.
Everyone ready to go? No? Just still got a pig in a bag.
Yeah, so I gotta bring it back. Okay.
They're ready, they're ready. Just type it.
What is this? See.
I think she's typing.
What she saying? We need another.
Okay.
Okay, that's fine. We're
No
just
problem.
waiting on one site to finish up real fast. All right, we are all set to go. So Kale's going to walk you through the dissection now.
All right, so we've got our pig here. We've got our dissection kit. Okay, you should have a pair of scissors, our scalpel, a sharp probe or two. You might have an angled one and a straight one, or both. Some forceps and a blunt probe. Okay, so those are the tools we'll be using. So if at any time during this you guys have a hard time seeing what I'm looking at or have any questions, please type us a question so we can get everything straightened out and make sure we're all on the same page. Okay, so the first cut we're going to make, we will be using our scalpel. This is the only cut we'll be using our scalpel for. Is our first incision. So the easiest thing to do to start, we want to find where the pig's rib cage ends. So if you can palpate down in the middle of the chest, and you kind of feel it drop off, and it gets a lot softer right about here. On mine, it's about where you see the first set of nipples here. So we're going to want to open the abdominal cavity right in that spot. So using our scalpel, we can start up a little higher. And so we'll cut down through the skin to the sternum. So we're not going to actually go into the thoracic cavity, but we'll cut the skin above it down to the sternum, making that cut down towards the back of the pig into the abdominal cavity. So when we get to about this area, be careful with the scalpel, but you can kind of open up your cut using one hand like this. You can see hopefully what you're cutting, and then try to cut down through the tissue until you see a small opening into the abdominal cavity. Put your scalpel down and look. Not quite there on this end. If you get a lot of fluid coming out when you push down on the abdomen, that means you're probably through into the abdominal cavity. Okay, and I can see mine there. So that's our first cut we want to make. So once you get into the abdominal cavity, you can put your scalpel away. And from here, for safety, we will use our scissors. We're going to want to make incisions from that primary incision along just below the ribs, down the side, and then we're going to cut down the middle towards the legs. So I'll help demonstrate that for you. I'm going to move my pig around to make it easier for cutting. Just want to find that opening in the abdomen, put my scissors in, and cut towards the side. As you open it up and peel it back, you can start to see some of the organs in the abdomen. Okay. Do that on one side. Try your best not to cut into the chest. Try to not go through the ribs. So here we can see we've got a flap opening up now. Now, we're going to take this primary cut down and go around the umbilicus here. So this is where the umbilical cord was attached. And we're going to cut towards the legs. Okay. So we should have him open kind of like this. You guys take a couple minutes to get to this point.
When you have all opened your abdominal cavity, just let us know and we will continue on with the dissection. We all set to continue on?
It's okay. Take your time cutting. Just try to get these couple abdominal flaps out. You'll notice if you lift up where the umbilicus is, there'll be a kind of a cord connecting from the liver to the umbilicus. These are our umbilical arteries and veins that pass blood from the mama pig to our little baby pig we have here. So you can keep that intact for now. We will cut it when we start the rest of the dissection and open up the other organs, but I wanted to point that out now as you might see it while you're cutting.
You can do that right now while you're just making them.
So as you're making these abdominal cuts, make sure they're nice and wide. You can open them up. If you get to this point, you can open them up a little bit wider. Let some of that fluid that's in there drain out into your tray. And then you can also cut the rest of the flap down here towards the hind legs. Really open things up. There's the pig's head on this end. Get him facing head towards the top of the screen. Help orient everyone. So once you get your abdominal cavity open to this point, this is kind of a good stopping point for us. We have our snack break scheduled for 5:30. So I totally understand if you guys are feeling a little hungry and want to do our snack now, you can come back and then dive into everything and not have to stop the rest of the way from here. Everyone sound good with that? Once we're done opening the abdominal cavity, we can take our break and have our snack.
So we will try to meet back by 5:40.
Is it? Yeah. Is everyone else okay with that?
Sounds great for the micro.
Okay.
Just type us if that sounds good to all y'all.
So we'll take our about 15-minute break now. All right, so we got some folks ready. Let's see. Our students are coming back in. Okay. Now we get into the actual fun stuff. All right, so one thing you probably noticed when you cut open your pig, there's some weird colors in there. These pigs were partially plastinated, so they used some different colors to inject in a few different vessels to kind of help you distinguish like some of the blood vessels, some of the veins, things like that within the body, within the pig. So hopefully you can see some of the different structures a little bit. So, we get over here.
You're on that ready. That girl is ready.
Okay. All right, so we're not quite ready. I could just keep talking a little bit though. So if you see inside your flaps of your abdominal cavity, you get this really shiny, you rub your finger on it, really slippery skin on the inside. This is called your peritoneum. So this is a specialized tissue that lines your abdominal cavity. It lines the outside wall, and it also lines all the organs within. So all of your intestines, your liver, your pancreas, everything is lined to some degree with this same peritoneal tissue. The purpose of this is to allow the contents in your stomach to move around a little bit easier. Like I said, if you feel it, it's really slippery. So that means when your organs are inside, they rub up against your abdominal wall, and there's not a lot of friction. So things don't tear or catch on anything. It also, as we'll see when we look at the small intestine, it kind of wraps around a lot of your organs and helps hold them in place. Because if your intestines were all just free and looped around, they could get tied in knots really easily. Then you'd have an obstruction and an infection, and it'd be lots of problems all the time. But because they're held together in an organized fashion, even though they are just long, hollow tubes, they don't very often get knotted up together. All right. Do we have micro lab? How are we doing? Awesome. Awesome. So, if you haven't already, the first thing you can cut, you can see here, is our umbilical artery and vein. They're wrapped together, and that's how nutrition passes from the umbilical cord to here, the fetal pig. As we pull this back, so we'll pull this section back towards the tail and kind of leave that back there. All right, so now we can use one of our probes. I would suggest using the blunt probe to start, if you've got it, and we can kind of start poking and prodding around. The first thing you'll want to look at is what divides the thoracic and abdominal cavities. That's your diaphragm. So if you look underneath here, it's going to be hard to see from my angle. I'm trying to tip him up a little bit. But if you look down in there, there'll be a thin sheet of tissue. That's a muscle called your diaphragm. It separates your abdominal and thoracic cavities and assists in breathing, which we will talk about a little bit in our next lab. So the first big organ you're going to see is your liver. It sits in the upper right quadrant of the body. Mine here is covered with a little bit of excess coloration, but it's going to be a solid organ. Oh, sorry, there we go. This organ here. It's going to be a solid organ. That umbilical vessel that we cut is going to lead into it. So that's this right here. Okay. Now, if you take the liver and lift up, what should you find underneath it? I wish I could hear you talking. Sitting on the bottom side of your liver is the gallbladder. So this is where your bile produced by the liver is stored. It can be tricky to see because it's very much, or it's like stuck to the bottom of the liver. So you might have to pick it off a little bit. I'm going to use my sharp probe here to try to separate it. Not sure you can see it very well. It's this little guy here. Kind of see it protruding out there. There we go. So this little empty sac right now is the fetal pig's gallbladder. If you look really close, you can see a little tube leading from the end of it, which will lead to the common bile duct and into the duodenum, where the bile will empty into the small intestine. To the left of the liver, and still a little bit behind it, will be our stomach. The stomach's going to be partially full. There's a little bit of amniotic fluid and some digestive enzymes that are most likely still in there. You can kind of feel it. It's a little bag-like structure, a little bit of fluid in it. And if you pull it back and look deep inside the cavity, you might be able to see the esophagus coming through the diaphragm and attaching to the top of the stomach. It'll probably be wrapped in a little bit of that peritoneal tissue, some kind of protecting it, but that tube at the top that leads into the stomach is your esophagus. In the thoracic cavity, you would see it sitting the very deep part of the chest, and again, we'll see that when we do a heart and lung lab. So if you follow the stomach, if you follow it down, this bag structure here, you follow it down and to the right, and you kind of squeeze at the end of it, you'll notice a big lump. Katie touched on this. This is your pyloric sphincter. So this is where the stomach empties into the small intestine, the first part, which is the duodenum. So this big tight spot is a big chunk of muscle that we'll look at in a little bit when we actually cut open the stomach. So leading from the stomach, we go into the small intestine. That's going to be the biggest mass that you see in the abdominal cavity. It's going to be a big wad of coiled intestine. This is where your primary nutrient absorption takes place. So if you spread it apart, you can kind of see, at least on mine, we have some yellow colorations kind of wrapped up in that mesentery. Those are the blood vessels that supply the intestine with nutrients, and nutrients that are absorbed from the intestines pass from those vessels and are absorbed by the body. The large intestine, you can differentiate. So the large intestine is a little bit different in the fetal pig than in humans. In humans, right, if you remember the picture, we have our ascending colon over here, our transverse colon coming across, and then our descending colon coming down. So it forms kind of an upside down U over the top of our small intestine. With the fetal pigs, they're coiled, the large intestine is a coiled structure as well, and it's sitting behind your small intestine. So if you pull your small intestine to one side, behind it, you should see a coiled set of tubes that is going to be a little bit bigger than your small intestine. And mine are also a little bit different color. It's a little bit darker green, and then the small intestine, which is more of a beige color. Does everyone remember what the first portion of the large intestine is? Be our cecum. And if you look around for it, the cecum is a blind pouch. So it'll be the portion of the small intestine that kind of goes nowhere. So right here with large intestine that goes nowhere, sorry. So here there's a little kind of like finger-like projection all by itself. So this would be our cecum, or the first part of our large intestine. Now, unlike humans, the pig does not have an appendix. So you can, if you try to look around for it, you're not going to find anything. What?
Can you slow down that hair?
Of course, of course. Sorry about that. So you take your time again with the small intestine. Large intestine behind it. Sometimes you might have to search to find that blind pouch of the cecum. I got lucky and mine was just kind of sticking out. If you've got a little excess fluid too, don't be afraid to dump it out into your dissection tray. Lost my ties. So far we've got our liver in the right upper quadrant, big solid organ. It's got multiple lobes. One lobe can be kind of tricky and it'll sit on top of your stomach. Then it's our stomach, the sac-like organ. Our small bundle of tubes, our small intestine. And then behind that, a little bit larger set of tubes, our large intestine. And then you might be able to follow it. So I can trace my tube here, and it comes down, and it's kind of behind that peritoneum now. I'll see a kind of a big green tube with mine going out to the anus. So this portion here would be our rectum that leads to the anus. Now there's one more accessory organ that we haven't looked at yet. That's our pancreas. So that sits pretty much right in the middle of the abdominal cavity, but way down deep behind most everything. So the easiest way to find it is to go back to your stomach, our big pouch here, and lift that stomach up. You can use your probe, and you'll have some skin coming off of it. Kind of tear that skin a little bit to release things. When you get behind it and start to cut through some of that tissue, you'll see a little little finger-like projection again, coming from the right side of the pig, or well, I guess your left, and coming out here to the right. It is wrapped in a little tissue as well, and it kind of looks like a little fatty structure. It's kind of lobulated and looks like little chunks all put together. And that's our glandular organ, the pancreas. It's not a very big organ. It's not a very strong organ. If you poke at it a little bit, you can see that it falls apart really easily. It's pretty delicate, but it's extremely important. It has a lot of functions, especially with our blood sugar control. And if your pancreas isn't working, which can happen with type 1 diabetes, and you have poor blood sugar control, it leads to a lot of other systemic problems. Can everyone see that pancreas sitting back there? Kind of blends in, at least from this angle, with our small intestine and our stomach, but hopefully live in lab, it's a little easier to identify. So that is like the gross view of all the structures that we wanted to look at. Now we're going to cut open a couple of our organs. Is everyone ready for that, or you want a little more time to identify the rest of the structures? You're ready. One thing we didn't mention in the PowerPoint, because it's not really a digestive organ, but as you were picking up your stomach, you might have noticed this little guy attached to your right side of the stomach, kind of opposite the pancreas. Kind of just looks like a little flap of tissue. This is the spleen of the fetal pig, and this is more an immunologically important organ. So it filters blood and removes any viruses or bacteria or other pathogens that might be there. And when your red blood cells get old, they go to the spleen and are recycled. So now we're going to cut open the stomach and try to see some of those rugae that we noticed in there. So everyone, use your scissors, and we're going to cut this lower portion, this lower curvature of the stomach. So you can make a small incision on one end to open it up, and then got a smaller end here, the sharper end, that you can kind of slip inside the stomach and make a nice cut and open the whole thing up. As you do that, some fluid's going to leak out. That's a cool view. So you might need to dump a little bit of that excess fluid into the tray. Okay. Now, if we open up our stomach, You can kind of see some rugae. So it's not quite as distinct as we showed in our picture in the slideshow, but you should be able to see a few small folds on that inner surface of the stomach. Like Katie said, those folds increase the surface area that's in there, allowing for better absorption and better breakdown of the food. More surface area is better for the intestine, so that's why you have these ridges pretty much throughout the intestinal system. In the stomach, they're called rugae. The small intestine, they are the plica circularis. You can also notice it looks almost velvety. So that's kind of your microvilli, or your your villi that line those rugae. I've got a kind of a cool view on mine. You can see I've got this big circular structure here. Looks like a big donut. So this is your pyloric sphincter. So this is the connection between your stomach and where it empties into your small intestine. So again, we can take our scissors. I like to use the small end again and kind of poke it inside that pylorus, inside that sphincter, and you can slice it open a little bit. Just kind of give you an idea of how thick. It'll be easier to see on your end again, but see how thick that muscle is. It's really important for digestive control because you don't want all the food you eat to just run through your stomach right into your large intestine. In your stomach, you have hydrochloric acid, and it really needs to sit there and be churned around in that acid to really help break it down into smaller and smaller bits. Because when it gets into your small intestine, if you have big chunks of food, your small intestine isn't going to be able to absorb that big chunk. It's got to break it down into individual molecules to absorb them one at a time. So that pyloric sphincter has got to be nice and strong and stay tightly closed until it knows to relax and let the food that's been thoroughly broken down in the stomach into the small intestine. And the last piece we will cut open for now will be a piece of small intestine. Doesn't really matter where, so just try to find your mass of small intestine. Try to isolate one little strand if you can, kind of like that. Again, you can see all those blood vessels going to and from it. Take your scissors again, and you'll kind of cut the tube in half. See that here? Like so. And then again, insert that smaller end into that opening that you've made. It's a little tricky. You might be able to use your forceps here to pick up your small intestine a little bit. There we go. Try to cut it open. So here on my end, I've got a little portion that I've cut open. You can't really see the plica circularis like we showed in the video. You might be able to see some foldings, but again, you'll notice that kind of velvety surface. So you know that are those villi lining your small intestine. More villi means more absorption, means more nutrients that are extracted from the food that you eat. So it's extremely important to have these villi. Otherwise, your small intestine, which is relatively short, gives it a lot of surface area and can absorb a lot of nutrients. So we're going to pause our dissection right here, and we're going to go into the urinary system. We got a short slideshow presentation that Katie's going to take us through, and then we will finish our dissection with the urinary system and anything extra that you guys want to look at. Do we have any questions so far? Everyone doing good?
Okay.
I hope that's a yes.
All right, so we are going to go do a run through of the urinary system. So your urinary system consists of your kidneys, your ureters, your bladder, and your urethra. First up, we have your kidneys. So your kidneys sit on the posterior wall of your abdomen, so behind all of your intestine. They receive blood from your renal arteries, and then the blood is then filtered to produce urine. And the portion of the kidneys that filters the blood is the nephron, which we will run through after we're discussing the overall function of the kidney. After the nephron produces urine, the urine moves through into the renal pelvis, which is collected from all the nephrons, because there's over 1,000 nephrons in one kidney. It runs into the renal pelvis, which collects all the urine to then be transported through the ureters. So here is a simplified version of your nephron. They sit within the medulla and cortex of your kidney, which I forgot to mention. The outer portion of your kidney is your cortex, and then the inner portion is called your medulla, and your medulla is sectioned into pyramids, which you can kind of see the broad base up against the cortex, narrowing down into calyces, which enter into your pelvis. Okay, so the nephrons, like I said, there's thousands of nephrons found in your kidney. They are the functional unit of the kidney, so they are doing all the work to filter out all of the waste, the ions, and the water in order to produce urine. So they receive blood from renal vessels that enter into your glomerulus right here, which is a bundle of vessels or a capillary. From there, the water, the waste, the ions, all that is taken out of the blood and travels through the nephron tubule. And as it travels through the tubule, the nephron is working to make it the correct concentration, take out the correct amount of water and the correct amount of waste and ions to produce normal urine. And the urine will then leave the nephron through the collecting tubule into the pelvis and then into the ureter. Before we get to the ureter, I just want to explain what you could we'll be able to see it on these kidneys, correct? The adrenal gland?
You should be able to. Yes.
So we'll be able to see the adrenal gland. The adrenal gland is not directly involved in the gastrointestinal or in the renal system, but it instead is responsible for producing mineralocorticoids, glucocorticoids, and androgens, which in an easy way of saying are hormones that help regulate your body. So those hormones are found within the cortex, so the outer portion of the gland, the adrenal gland, and then the medulla produces epinephrine and norepinephrine, which are hormones that are part of your fight or flight system, which some of you may have learned already what that is. We won't go into a ton of detail about it, but it just increases blood pressure and heart rate and gets you ready to make a correct decision, or makes you ready to run away or fight against a bear. It gets your adrenaline pumping. Okay, so back to the urinary system. Like we said, from the kidney we go to the kidney, the pelvis of the kidney, pardon me, and then the urine will be transported into the ureters. So the ureters exit the kidney at the same spot, actually, that the renal artery and veins enter into the kidney. So there's one spot on the kidney, it's called the hilum, where things exit and enter the kidney. So the ureters will exit there and travel down the posterior wall all the way to the bladder, which it will enter onto the back side of the bladder. And then finally, you'll be able to see the bladder. I'm not entirely sure if you'll be able to see the urethra, but the bladder stores the urine produced by the kidney, and it expands until it's completely full.
Human bladder, yeah, probably.
And a human bladder can hold about 1 liter of fluid, and then when it can't hold any more, the bladder is actually it's a organ that, just like the stomach, the walls are covered by muscle. So it contracts these muscles and it pushes the bladder out through the urethra, which is the tube that connects the bladder to the outside. So that is actually it. And so now Kale's going to show you the kidneys. Okay.
Okay. All right. So we're here for the last little bit. So did anyone see the kidneys while they were looking around earlier? Hopefully you did. You might have seen them when you pull those small intestines to the side. Got our large intestine there. Pull the large intestine. You've got that peritoneum sheet, but behind it you might be able to see a little outline of the kidney. Okay, so in humans, the kidney sits here, and your adrenal gland sits kind of on top of the kidneys. But in these fetal pigs, the adrenal gland sits more towards the front and kind of in the middle. So it should be more over here. Might be hard to see. It'll look kind of like the pancreas did. So we have kind of like that grainy structure that looks pretty fragile. It's going to look very similar. So what we want to do is we can use, I think the shark probe works well here. Go, and you just want to tear through this lining covering the kidney. Again, this is your peritoneal lining, and your kidneys are kind of stuck behind them, held in place by that peritoneum. So once you get that open, you can use your blunt probe to poke around a little bit. So here, underneath this capsule, this surface here, I've kind of got some of that soft, lobulated material. Try to zoom in a little bit. Camera can focus now. Might be too close to focus. If I had some Google glasses, this would be perfect. So, anyway, you should be able to kind of lift your kidney out and see that, oh, got a little excess fluid again. Squeeze and try to get some of that out. But with that coloration, you might be able to see some of those vessels leading into the hilum. Okay? So you want to separate your kidney, pull it out, and using your scissors, go in and cut those vessels leading into the hilum. We are left with our kidney. Okay. So here's the hilum that we cut. Let's see if we can get it to focus. Some of that granular tissue right here. But now we're going to cut our kidney open, and you can see the medulla and the cortex and see how they look different. So the easiest way to do this, I think the safest way, would be with our scissors. So we want to cut, we've got our hilum here, where our vessels go in. It's hard to see on this picture exactly, but the hilum's right here. So we want to cut in line with the hilum, okay? So you can just kind of hold your kidney, use your scissors to cut it in half. Mine did not. Not the perfect example on this end. You can kind of see on mine, you can't see the pyramids perfectly, but you can see there's a difference in the tissue. So there's one tissue that kind of goes around the outside. And so that would be our cortex. And then the tissue in the center is a little bit different. And that is our medulla. Once everyone gets a good look at those, we'll go back to our pig. Take your time with that. No need to rush. Okay, once everyone is done checking out that kidney, if you are, you might still be working on it. Are you still working on the kidney? I don't want to go too fast. Not hearing anything or seeing anything. If we go back to our pig and flip to the other side, we'll take a look at that kidney now, since we cut out the first one. So we can open up that again. With our sharp probe. Use our blunt probe to kind of pull everything away. Now we're going to look for the ureter. So that should be a small tube leading from our hilum. So our urine is collected in our renal pelvis and goes out the hilum in the ureter. And that'll be a small muscular tube right here. Trace it here. leading down towards the bladder. Now, on the pigs, well, so in a human, you normally see the bladder sitting right here in the pelvis. Let's get this up a little bit. It's normally sitting right here on most human anatomical images. But when we made our cut with the umbilicus and flipped it back, you might have noticed a sac-like structure on the inner surface of that little flap that you made. It's got a couple arteries running over it that head towards the umbilical cord. Okay, so those are our umbilical arteries. But this sac behind it is actually our bladder. And if you follow it back, you should be able to see where our ureter comes here. Ureter comes here and dives into this back side of the bladder. A lot of images that you'll see of bladders also show them nice and inflated. So they seem really easy to find, but in reality, they're usually empty. So they're going to be kind of little collapsed sacs. So this is really the last thing we have to cut here today. And we're going to kind of do like we did the intestines. We're going to cut it in half, and we're going to cut it down the middle a little bit so we can open it up, see how thick that muscular wall is, kind of open up the bladder, okay? So just take our scissors here, make an incision like so. You can even get your probe in here a little bit. See how it stretches open and enlarges. Okay. And if you want to get a little better look, use your scissors again to transect it longitudinally and open it up. So when you think of the bladder, it's easy to think sometimes as though it's just a little sac, but it's actually got a relatively thick muscular wall around it. So when you really have to go, that muscle squeezes and really empties the bladder quick. Does anyone have any questions about anything that we've done or talked about or that you've seen? Maybe any weird things you saw in your own pigs at your site? Any questions?
I'm gonna mute everybody now.
I should know which either pig that goes. I will be really done. Minus. He's ever done, but the plate will be right. If we found anything that we wanted to go through, I found a lot. Have at it, go ahead. Ew!
I get the brain.
No, that's gonna be hard. Yeah, we try to get them. Where do you want us to put the tools?
Okay, anyone who told you to scalpel, I should have had this told you this before we started. You're gonna hold it like a pin and make sure you cut away from you. I've had a student slice off the top of her finger before, so I don't want to repeat of that. Yeah, I had to call my.
I skip over this one all the time. Really doesn't matter.
So we hope everyone enjoyed the lab today. Hopefully you had a successful dissection on your end. I know the video is probably a little bit difficult at times. I wish I could show you guys some better angles. a few of these things, but hopefully you got to see them on your end. We've got 3 more of these labs scheduled through the year, so hopefully you guys will take part in those as well. Our next one we're going to do
Cardiopulmonary.
yeah, we're going to do a cardiopulmonary. I think we will be opening up the chest cavity of these pigs and checking out the stuff going on in there. That is the current plan. So hopefully you guys can join us again.
Yeah, and then in the late winter and early spring, we're going to be doing the oral cavity and dental, and then we will also be doing
neuro-related one.
Neuro-related lab.
So more to look forward to all year.
If you could remember to now take the post-lab quiz, which is the same quiz as the pre-lab quiz, but it's just to see if you hopefully that you learned more from this lab and this dissection. And then also, I believe there is a post-lab survey to see how your time was and what other recommendations we can make.
Any improvements we can do for next time.
All right. Well, thank you, and we hope you all enjoyed it.
We're done.